This short video will explain the experiments we have planned for the next 2 weeks, what we're trying to achieve, and what I want you to learn. There are 2 sets of experiments. The first set will aim to determine the regulation of an alternative splicing event which has clinical relevance in the disease aniridia. The second set relate to the separation of blood and the analysis of the proportion of the different mononuclear cells within a sample. Assuming we have no major difficulties, the first set of experiments will form the basis of your write-up, and so I want to focus this video on the different parts within this set of experiments so you understand what's going on. First, some quick background. When you first learn about DNA, RNA and proteins, you get told that one gene gives rise to one messenger RNA, one mRNA, and that the mRNA is then translated into one protein. However, it's a bit more complicated than that. Each gene can produce multiple different mRNAs through a variety of different processes, and the one that we care about is alternative splicing. So most genes in higher organisms contain stretches of DNA that provide the code for the protein. We call these the exons, and in between these, the intervening sequence doesn't code for proteins but is part of the gene, and we call these regions introns. Now ultimately, the final mRNA is made up of just the exons. So there are various different ways that alternative splicing can happen, but to keep this simple, we're only going to consider a simple cassette exon. For cassette exons, depending on the conditions within the cell and at the time the mRNA is being made, the exon may or may not be included in the final mRNA. The result is that there are 2 or more different versions of the proteins that can be made, a version with the cassette exon and a version without. The resulting proteins can be structurally and functionally distinct. In our experiments, we're investigating the transcription factor PAC6 and a splicing event that gives rise to either PAC6A or PAC6B. These 2 alternative splice isoforms are closely related to one another, but play different roles in ocular and pancreas development. Their relative expression levels, i.e. the ratio between the 2 isoforms, is tightly regulated, and defects in that splicing regulation are associated with pancreatic cancer and ocular surface disorders. Our experiment will investigate the genetic elements that determine which isoform is produced. Our aim is to determine whether which parts of the native PAC6 sequence control the splicing rate of the cassette exon. Now, my group have studied the gene in detail and have identified some regions that we think are likely to be key to regulating the splicing switch between the different isoforms. Our overarching hypothesis is that by mutating some of these specific sequences, we'll get either more or less splicing, i.e. we'll change the PAC6A to PAC6B ratio. I'll come back to the specific experimental hypotheses shortly. So, how are we going to study this? Now, we could use genome editing to introduce mutations into cells, but that would take a long time, be quite expensive, and is not really a justified expense when it comes to screening lots of different mutations. Instead, my lab have made a dual colour minigene to allow us to ask our questions in a more sort of high throughput way. The minigene has 3 parts. On the left, it has the 5' end, it has the sequence encoding a red protein, M. cherry. On the right, the 3' end, it has the sequence for a green protein, GFP, and in the middle, it has a specific stretch of sequence from the PAC6 gene, which includes the cassette exon, exon 5B, that is included in PAC6B, but not PAC6A. However, we have made one important change in the sequence. In the cassette exon, we've introduced a stop codon in the middle. This means that any mRNA that includes this exon will stop translation at that point. Now, we've designed the minigene in this way so that it gives us a colour readout of the situation within the cell. When we have splicing that skips exon 5B, we'll produce a protein that has the code for the red protein and for the green protein. However, when we have situations where exon 5B, the cassette exon, is included, then we'll produce only a red protein, and that red protein will actually be smaller than the green protein. Note that this means that every cell that has got the minigene has got the red protein, and it's actually the ratio of the green to the red protein that we're going to care about. They'll tell us how much splicing has happened. Okay, that's the baseline, but our experiment will ask what effect different mutations have upon the splicing rate. My lab has made a range of different mutations. Which ones you'll be using will be explained to you in the lab. Make sure you take a note of this because we do different things each year. So here's what's happened. We've taken cells in culture and we've got 5 different conditions. 1, cells with nothing added. 2, cells with our wild type minigene added. 3, cells which we have made make the red protein only. 4, cells which make the green protein only. And 5, cells into which we've introduced our mutant minigene. So this was all done ahead of you by people in my group. The experiment then has 4 parts. In part 1, we will use fluorescence microscopy to take images of the individual cells. This will be a quick check to make sure that we have green and red protein being expressed in the different conditions, and will give us a sort of first indication of if our mutant has an effect or not. In part 2, we will run our cells through a flow cytometer. This will measure how much green and red fluorescence is in each individual cell of the population and will allow us to determine a cell-by-cell green to red ratio. We will do this on 3 independent experiments in the lab. It will give us quantitative data that we can then perform statistical analysis upon. In part 3, we will analyse which proteins are produced in each of the conditions using Western immunoblotting. This will tell us the size and the relative abundance of each protein in each of the different cell conditions. It will confirm that when we saw red or green fluorescence by microscopy or by flow cytometry, that it really was from the protein we predicted it to be from. Part 3 actually has 2 steps. First, you will determine the protein concentrations using a Bradford assay, and then second, you'll separate those proteins by the size using sodium dodecyl sulfate polyacrylamide gel electrophoresis, SDS-PAGE, and then you'll perform Western immunoblotting with antibodies against the green protein and the red protein to detect their relative size. We'll be able to quantify these blots, which will provide validation or triangulation with our flow cytometry data. Finally, in part 4 of the experiment series, we will analyse the mRNA produced in each of the different cellular conditions. This will involve taking RNA samples, using reverse transcription to convert it to complementary DNA, then performing a polymerase chain reaction, PCR, to determine which products are present in each sample. This will confirm that the proteins have come from the mRNA that we predict and will help identify the reason behind any unexpected findings that we might get. After you complete these experiments, you will analyse the data and write up a report in the style of a manuscript or a scientific journal article. And I'll explain this in a separate video or lecture. Don't worry if not all this has made sense right now. Things will become a lot clearer as the week goes on and we explain each of the different steps of experiments, and there'll always be chances to